Thank you. Well, good morning or good afternoon to everyone joining us today. So today we'll be hosting the October session of the Amplifying Voices webinar. My name is Bianca Rodriguez Cardona, and I'm one of the ASLO Early Career Committee members. For those joining us for the first time, this webinar series has the purpose of promoting the work and research of early career researchers from underrepresented and historically marginalized groups in our global community. If you're interested in presenting your work in this webinar series, we will share the application link in the chat and everyone is welcome to apply. And today we will have two very interesting talks from microplastics in streams to mountain freshwater crabs. And so we are all aware of how today is going to work. Each speaker will give a 20-minute presentation followed by a quick 5-minute question session. And then the second speaker will speak for another 20 minutes, and then we'll open the floor for both speakers to get questions and discussion time. So today we will start with Dr. Frank Akamagwuna. He's a postdoc at the Institute for Water Research at the Rhodes University in South Africa. And he will be giving the talk titled Microplastic Proliferation in River Systems. Can they support the establishment of macroinvertebrates and emerging pathogens? So Frank, please take the floor.
Thank you, Bianca. I'm just going to quickly share my screen. Excellent. I hope the screen is up and running.
Yes,
yeah,
we
looks
can
good.
see.
All right. Thank you, everyone, for, good morning, good afternoon, and good evening, wherever you're joining us from today. And a very big thank you for making our time to participate in this presentation. I'm actually excited to be talking to you today about macroplastic proliferation in river systems, asking the question if they can support the establishment of macroinvertebrates and emerging pathogens. So my name is Frank, like Bianca said, Akamagwuna. So I'm from the Institute for Water Research. The Institute for Water Research is in the eastern part of South Africa. I'm going to be talking to you today alongside Andrew Ali. Andrew Ali is actually a master's student that is also actively involved in the project. So Andrew is going to be chipping in here and there during the course of the presentation. Maybe you say hi to your audience.
Hello, everyone. Thank you so much for having us today. We're really glad to be part of these interactions today, and we hope that we all have something to take home with at the end of the interaction. Thank you.
All right. So, and of course, the project PI is Professor Nelson Odume. The research work is actually conducted in his lab. And without further ado, we'll just head straight to the main presentation. So background, initially I had wanted to, the initial plan I thought was to give a kind of a brief overview of the kind of work and the project, the research work in which I do, and that we conduct in the Institute for Water Research. But I was made to understand that time is not that friendly in terms of the time allocated for the presentation. So I'm just going to give an overview of the kind of research I've been working on, then delve into plastic pollution, which is the focus of this presentation. So freshwater pollution. Freshwater pollution actually represents the most threatened ecosystems. And the greatest threats actually comes from multiple anthropogenic pressure or stressor, as the case may be. And some of this pressure actually spans across agricultural activities, climate change, urbanization, sedimentation, and of course, recently emerging pollutants. And what are the consequences of these activities? A major consequence of these activities in river system actually spans through poor water quality, decline or decreasing availability of freshwater resources, especially in the part of the world that I'm from. Africa has only faced a lot of problem with regards to availability of water. A major issue that comes from this range of anthropogenic activities is actually biodiversity loss. There's a very massive decline in biodiversity. There's also a depletion of ecosystem services as a result of the a range of these anthropogenic activities on freshwater ecosystems. And of course, the lack of access to water is a major concern that actually resonates from these activities. So as a result, most of the work I've been involved in till date has actually focused on exploring and developing various approaches and tools for monitoring and assessing the effect of multiple pressure or multiple stressors on freshwater ecosystems. And of course, recently, I'm now focusing more on emerging pollutants with a core focus on plastic pollution, which is actually the main, the core focus of this presentation. So plastic proliferation. So when we talk about plastic proliferation in the river system, the major culprit to this is actually the single-use plastics in river systems, which actually range from your plastic bottles, your packaging materials that are used in packaging and all of that. So these plastic materials are actually single-use plastic that you use and you dispose them. But the problem is that most cases when we use these plastics and we dispose them, most people don't even know what the ripple consequences of disposing some of these plastic, especially when it comes to developing countries. But the problem now is about 400 million tons of plastics are actually produced annually, and less than 10% of these plastics that are produced are recycled. The question then is, what happens to the rest of the plastic that are produced? And with the current historical trend actually shows that the production of plastic is actually expected to increase by 1,000 and above 1,000 million tons by 2050. And this is actually worrisome, especially in developing countries where, like Africa, where population growth is also projected to double by 2050. And more recently, more than 50% of plastics actually ends up in the environment. The question then becomes, when they end up in the environment, where do they go to? The major place they end up is in the river systems. And when they're in the river system, they then cause a range of direct and indirect socio-ecological problems, ranging from direct poisoning of aquatic organisms, reduction of aesthetic values of freshwater ecosystem, and a range of other problems that they causes. But recently, plastics themselves, which are macroplastics, they have been shown to provide artificial habitats for organisms, which is the concept they now refer to as platisphere. So, but the question now is, when they provide habitat for aquatic organisms in river system, what are the implications in terms of ecosystem functioning and the ability of freshwater ecosystem to continue to sustain itself? So a range of problems that they cause is the distortion of natural biodiversity distribution, the distribution and transport of invasive biota or invasive species, which actually has a range of implications in ecosystem functioning, including aquatic food web. They can also be facilitate or be vectors of pathogens, which has some public health implications, such as disease transmission, because people actually depend, when you come to this side of the world, you then realize that people also directly depends on this river system for their direct, for their daily survival in terms of drinking and other uses. So there's a concern of public health risks. But however, the problem then is, when we talk about plastic research, most research are actually focused on microplastics. And that then leaves us with a limited understanding of the effect of the bigger plastics. So that is where the main focus of this project is actually focused. So the question we are then asking, if plastic becomes the primary habitat in river system, what are their implications in this case? So that then brings us to the aim of the study. So the aim of the study is to investigate the influence of biohabitat complexity on the dynamics of macroplastics and biota assembly distribution in rivers. and as well examined whether microplastics can support the establishment of macro and microorganisms, including emerging pathogens. So this is actually the overall aim of the study. And the specific objectives are actually highlighted here, but I'm going to just lay more attention on the first objective, because for this study is actually giving us a preliminary understanding, because it's an ongoing project. So we are going to be presenting preliminary results that are actually coming out from the project. So the specific objective we are going to be focusing on is to assess the assemblage colonization of macroinvertebrates on macroplastics in selected river systems across a range of land use activities. So method. So what did we do? So the first thing we did was to select our study area. So we selected area. Initially, we selected 8 study sites in 4 river systems that range across different anthropogenic activities, including your urban activities, agriculture, and of course, informal settlement. But then we realized that we first have to establish the fact that before we can then begin to interrogate the different colonization across different land use activities, we then felt that it is necessary to actually establish the fact that, or establish if these plastics can actually support colonization of biological assemblages. So we then decided to select 4 sites within headwater streams of 4 river systems in the Eastern Cape, which include the Swartkoop River, the Cat River, and the Blokrans River, and the Buffalo River. So we selected 4 sites in 4 river systems, and we actually selected sites in the headwater streams of these river systems. And the next thing we then did was to conduct a field colonization experiment in these headwater streams to actually establish if really plastic can be able to provide habitat for biological assemblages. So that would then lead me to the next part of the presentation. And what we then did here was to actually come up with a gradient of plastic substrates. And I will invite Andrew to actually lay through more insight, because he has been actually working actively on this aspect of the project. Andrew, over to you.
Thank you so much, Frank. So basically, we did a field experiment to look at how our plastics are able to be colonized by macroinvertebrates or biological assemblages overall. So now it has been established that in our river systems, plastics do entangle with the aquatic plants, which are your macrophytes, and also with the sediment and different river structures like the boulders and your pebbles and all that in the river system. So for us to be able to establish how these things work, because we have research that have established in a way that these plastics harbor biological assemblages. But because we needed to understand the framework of time, the timing and all that, because most research will give you that, yes, there's this habitat that plastic is establishing, but then there's no really, they can't really tell that for how long has this plastic been in this place. So that's why we did this experiment. And so we came up with 3 different substrate types. One, which is the natural substrate, and we call this natural substrate the NS, just where you see NS. And then the other substrate we have is a mixture of natural substrate and plastic substrates of equal proportion, which we have as NP. And then we have the plastic dominated substrate, which is basically purely plastics. Now, the issue of having the natural and plastic substrate together is because since it has been established that these plastics entangles with the other habitats, like your boulders and the plants and all that, and the debris is in the river system. So we have this. Then we're also trying to look at what happens when we have a river system that has completely plastics in it. There's nothing else, although there will be something else on it, but these plastics, when they overshadow that habitat, what will happen? So that's why we're trying to see how would this be differentiated in the long run. So for our experiments, as we said earlier on, we had 4 river system. And alongside this 4 river system, from the beginning of our deployment to our retriever, consequentially, we always took, we're also taking water quality parameters, which are your environmental variables. And then for the field experiment, we try to make sure that we because the river habitat, the river system comprises of different hydrological biotas. So we chose 3 different hydrological biotas, which has to do with your runs, the refill, and the pool. These hydraulic biotas are differentiated based on their physical look and the rate of flow of the discharge of water from those systems. So our plastics were deployed in all different biotas. And then after we took, every month, we went back to the river system to pick up our deployed substrates, which we did monthly for the period of 6 months, that is 180 days. And then we counted our invertebrates from each substrate. Now, in the laboratory, we also did nutrient analysis, where we look at the different nutrients in our water system, just to be sure that this river system that we took are alike, and they are all headwater streams, they have not been polluted. And then for now, in this presentation, we're only going to be presenting preliminary result on the macroinvertebrate identification and analysis that we have been able to do so far. And then going further, we'll do more analysis to look at the trade analysis and also the sequencing for the other assemblages. So Frank is coming back to continue with the results for now.
Yeah, thanks, Andrew. So what Andrew was basically saying, that we deployed the plastics, and they were retrieved monthly over a period of 6 months, which is 180 days. And so I'm just going to quickly, for the sake of time, go through the preliminary results that are coming out. So because we're actually focusing on the preliminary result of macroinvertebrates, we are currently doing analysis on the pathogens and the rest of the other aspects of the project. So in terms of preliminary assemblage of macroinvertebrates across the gradient of plastic substrate, so one thing that is already coming out is that the natural substrate and the plastic dominated substrate, including the plastic substrate itself, that is the plastic dominated substrate, are actually having the highest abundance of macroinvertebrates, including macroinvertebrate richness, as opposed to the natural substrate, as you can see from the figure. So this is actually natural 50 equal proportion of natural substrate and plastic substrate. And this is natural substrate, and this is plastic dominated substrate. So you can see that the natural substrate actually has the least abundance of relative abundance of macroinvertebrates. And again, it is also very important to note that this colonization of plastic substrates were actually largely influenced by the type of hydraulic biotopes that we are present in the river system. That's the different section of the river. So the colonization potential we are actually influenced by the hydraulic biotopes. And again, so this hydraulic, the macroinvertebrate colonization actually declined from your run to your refill and to the pool. So basically the run, which is the running water, actually that biotope actually had the highest abundance of macroinvertebrate, and of course the pool had the least abundant. So here we were thinking, okay, what could have been responsible for the influence of hydraulic biotopes on the potentials of macroinvertebrate to actually colonize plastics. And so we decided to just give you a kind of a preliminary idea of the water quality in between or across the different hydraulic biotopes. And so what we noticed was that nutrients themselves, the nutrient variables actually didn't differ largely across the different hydraulic biotopes. But then if you look at DO, you would then realize that DO actually statistically differed across the different hydraulic biotopes. For example, the run actually had the highest amount of dissolved oxygen. And of course, velocity also had, there were more, of course, this is actually expected. So there were more velocity flow in the run. So we were then thinking that the different water quality as you go from one biotope to the other may have a very huge influence on the ability of macroinvertebrates to actually colonize plastic substrates. Then we also tried to look at the temporal assemblage across the length of the experiments that we conducted. So we then also realized that as you go from the first month to the last month of experiment, the colonization of plastic substrates actually differs statistically, where you then have, but then these differences differed between the different substrate types. So for example, in fact, generally macroinvertebrate abundance in the plastic-dominated habitat actually increased over time, whereas the abundance for the natural substrate we are seen to gradually decline as you go from month 1 to the last month of the experiment. So, and again, this trend was also influenced, or this colonization potential across the month of deployment, we also influenced by hydraulic biotopes in the study. So, so this is the assembly structure of the macroinvertebrates across the substrates. And now I'm also going to invite Andrew to share a little bit of insight on the results that are coming out from here.
Yeah, so thank you, Frank. So basically, we just try to look at how these assemblages were as a whole, like a community for each substrate. And then we try to find out from the hydraulic biotopes to see how these things, but then we were not able to see a lot of differences. But then we did a little ordination here, which is the PCA ordination, which gives you a relationship between an effect and the kind of response it gets from the organisms around it. So here, basically, we tend to find out that from our analysis here, we found out that at the beginning, when you look at the top left side of our plot here, you find out that all the substrate that we had in month 1 had this similar set of organisms, macroinvertebrates that were colonizing them, and the best part of it is that we found that the sensitive EPT at some point in time were showing a bit of negative relationship with our plastic-dominated substrate, which, if you look at this towards the top right, you find out that macroinvertebrates such as the Acevedo-Alonso and the Simulidae were more associated with our substrate towards the ending of our experiment, which is towards the 4, 5, and 6 months. They were not really there from the first month. Now, we also look, when you look at these things, now we try to understand going further when we start to look at the trade of these organisms, to look at what is it that's enabled them to come to be able to colonize this substrate towards the ending, and why were they not there in the beginning? These are some of the things we'll be looking at in the future.
Thank you, Andrew, and yeah, thanks for actually presenting that very carefully. So, so I'm just going to quickly go to the conclusion of the study. So generally, one can then say that microinvertebrate colonization actually changed across the plastic gradient, with plastic-dominated substrate actually having the highest abundance of macroinvertebrates. But however, the assembly structure of macroinvertebrates across substrates and across the period of deployment, we are largely influenced by the stream biotopes. And again, this macroinvertebrate colonization were also largely taxa-specific, which is what Andrew was actually saying earlier. So the colonization potentials of macroinvertebrates were actually dependent on the individual characteristics of each of these macroinvertebrates, which are some of the things we are going to be unpacking as we go along the project, then looking at the individual or the intrinsic characteristics that actually enabled a specific taxa to colonize plastic and dominated substrate. And so overall, plastic substrates supported macroinvertebrate establishment and may be mediated by like I said earlier, the special traits that these organisms possess. So we then hope that further work and analysis should be conducted to actually unpack some of the preliminary findings that are coming out from this study. And of course, we're also going to do the microbial sequencing, which is actually what we are working on currently, to unpack some of the effect of or the implication of if plastic then becomes the primary habitat in river system and they're able to transport or establish microorganisms or concentrate aquatic microbiota, what are the implications in the context of Africa, where people actually have these direct linkages and interaction with freshwater ecosystems in Africa. So what are some of the limitations in the study? I mean, some of the two major limitations I've decided to highlight here is the issue of interference with the field experiment and the flooding. So, I mean, sometime when we get to the field, you realize that the experiment that we have deployed in the field have actually been removed by people and kept them there for us, or remove them and take them away. So those are some of the major challenges we faced during. So the challenge actually affected some of the outcome of the result that are coming out. And again, flooding event as well. I mean, the deployment happened during the period of flooding events that actually took place, if I remember, July or thereabouts in South Africa. So there were a lot of high flow events that washed away some of the deployed plastics in the field. So those were the major limitations that we noticed in the field. Then acknowledgement. So we wish to acknowledge the African Multiple Cluster of Excellence for funding the study, and of course the AWAM, African Resource Mobility Network, for the funding support as well, and of course Rhodes University for providing the platform for us to actually conduct this study. And I would then say thank you to everyone for making our time and for listening to us today. And we hope to entertain questions and insights. Thank you.
Thank you.
Okay, thank you very much, Frank and Andrew, for this great presentation. We have time for a quick question for Frank and Andrew. But a reminder that towards the end, we'll have much more time. We'll have time for more in-depth conversation and questions for them. But for now, we can take a quick question.
Bianca, there's a question in the chat.
Yep. Haley, do you want to ask your question, or I can read it for you?
Sure, I can ask it. That was really interesting. Thank you both. I just had a quick question about whether or not you found that the plastic treatments trapped a lot of sediment. I guess I'm curious to think about if you think that the higher abundance or diversity in the plastic treatments was because there was additional resources for them there, like they were trapping natural debris, or just purely the physical habitat, like the increased surface area was perhaps why they were there. Just curious.
Thank you. Do you want to come in?
Yeah.
Yeah, Andrew is keen to light to that question. All right. Yeah.
So
go for it.
Basically, you know, we from our observations, we tend to find out that it's obvious that the presence of these plastics in that water in the river systems brought about the growth of a lot of biofilms and all that. And most of our organisms, like your shredders, which look for food. Now, the plastic in itself comes with a lot of surface area compared to, which is a bit complex than the natural substrate that is in the river system itself. So this already, and I think that this is one of the supports that the hydraulic biotopes had, that made it had a lot of organisms, which we know ecologically that normally that biotope supports more organisms than the other biotopes. But then the presence of these plastics, the surface area there, and then the presence of the biofilms also were some of those things that contributed to the high abundance of organisms in our substrate.
Thank you, Andrew. And I'm not sure if there's another question.
Yeah, well, I think, well, we can ask some follow-up questions to that afterwards towards the end of the session. So we will switch off into our next speaker now. Thank you, Frank and Andrew, for your time and your presentation. So we can return back to ask them more questions at the end. So now we'll switch speakers. So next we will have Ada Acevedo-Alonso. She's an independent researcher and remote ecologist in Colombia. She's also a carcinologist. And today she will be talking about the conservation status of the Neotropical mountain freshwater crab. So go ahead, Ada.
Okay, so good morning. So I'm gonna share my screen so you let me know. So I think we are fine, right?
Yep.
Yes.
Excellent, excellent.
Okay, so as Bianca says, I'm gonna talk to you about the conservation status of the Neotropical mountain freshwater crabs. That's the superfamily Pseudothelphusoidea that used to be the family Pseudothelphusoidea. You know a little about freshwater crabs, so that's a work that I did with Neil Cumberledge. He's from Northern Michigan University, but I'm here by myself. So we are gonna talk a little about freshwater crabs in general, where they are and their importance, about Pseudothelphusoids itself, how you do a conservation assessment, the reference assessment, our results, and our conclusion. So first of all, freshwater crabs. So we have around 1,500 species in the world. They are in 122 countries, and they are 6 families described. So we have the first one, that is Photamidae, that lives in Europe and Asia. The second one, that is the Carcinologist, that lives from Asia to Australia. The third one, that is Pseudothelphusoidea, that lives in the tropical region of Africa. And the other 3 ones are here in the Neotropics. So it's Trichoactilidae, Epilobocerea, that is restricted from the Caribbean islands, and Pseudothelphusoidea. So these 2 last ones are the main objective of the study. So here is a map of their distribution. So it's in numbers of species by countries. So if you notice here, we have 2 hotspots that will be China, with being the most biodiverse country in terms of freshwater crabs, and here Colombia, that is my country. We have kind of the 3rd percent of Pseudothelphusoid fauna, so we have a lot of crabs. So what is important of the freshwater crab species? So they are considered a key species, so they can represent up to 80% of the biomass in freshwater systems. So they are prey of many animals, birds, monkeys, other mammals, fishes. They are also cannibals, so they are prey of other crabs. And they consume a lot of animals, so macroinvertebrates, frogs, lizards, fishes. They also eat macrophytes, and they are important in the nutrient cycle because they process the sediments in the freshwater system, and they eat a lot of meat litter. And they are known as water quality indicators because they are really sensitive to pollution and thermal changes. They are also important in human health. So there is 2 diseases that are important with crustaceans involvement, especially freshwater crabs. So the first one is the human lung disease. The paragonimiasis is present in tropical places, both in Africa and here in Latin America. So the crab carries the metacercarine of this trematode. And if a mammal or a human eat a raw crustacean or an undercooked crab, they get infected. And some cases are known to infect liver and brain. And the other disease is the river blindness that is present only in Africa. So here the crab is not a vector itself. The vector is a black fly, but the larva of this black fly is carried by the crab. So when the crab goes inland and walks or is in the river, is carrying the larva with the nematode. So it's dispersing the vector. And they are also important in culture. So they are now a good food source for local and indigenous communities. They are involved in some beliefs. So for example, here in Brazil, there is an indigenous community that use a lot of freshwater crabs, and they have a special ritual for girls. So they have like a couple of days that they are only eating crabs because of their religious belief, because of their nutrient value, and also in local costumes. So here in the picture is a crab. So this is a clay pot from an indigenous community here in Colombia. So hopefully you can see here are depicted the claws of the crab. Here's like the rostrum that will be like this part of the crab. And here down is the abdomen, that is the same up here. So just an example that they have been in culture and they have been noticed since a while ago. So Pseudothelphusids itself are comprised by two families, as I told you before, Epilobacteriae and Pseudothelphusidae. We count now for 294 species, but this study only covered 293. The last one was described like last month, so it didn't make it to do the study, but so you know, we have a new species and they are distributed in the Neotropical region from Mexico to Brazil. So they're associated with mountain ranges. So here all the yellow points are all the freshwater crab of the Pseudothelphusids that we have recorded. So they go from here in Mexico, from the Sierra Madre mountain range, here in Latin America, in the Andes, and here in the Guyana and Brazil highlands. So they're associated to mountain ranges. That's what they are called, mountain freshwater crabs. So they live in a high altitudinal range, so they can go from 400 to 3,000 meters above sea level. They are semi-terrestrial, so that means that they live both in water and inland, and they can breathe air. So majority of the freshwater crabs can breathe air, but this Pseudothelphusids has a special structure that is called pseudolung. So the tissue has a special modification that allows them to take oxygen more efficiently from the air, so they literally can breathe air. They have direct development, that means that they don't need to go under salt water to breathe, and they have parental care. So if you see, this is a little mama crab from Mexico, and all the things that you are seeing here are little crabs, so they have parental care, and they inhabit clean freshwater bodies from creeks, streams, lakes, swamps, caves. And there's a nice example in Costa Rica where a crab is specifically living in leaf axils, so he is not using anymore like a freshwater system like a rivers. It only depends in the water that is in the leaf axis. So that's another one too. So here is a little video just for fun. I say it's a little crab mama with her baby that's here in Colombia in a cave. They can live also in caves. So as I told you, they are around 300 species and 256 of them are country endemic. So they are only present in that country. So we have a high endemism rate, and that's because of the altitudinal range. So they have a specific distributional range that is associated with an altitudinal pattern. So we have described 48 genera, and of that 48 genera, 99 are monospecific. So only one species have been described. So before the actual study is only being done one assessment that was done in 2008. And they found that around 14% of the species of Pseudothelphusids were threatened by extinction. There were some local assessments. In 2015, there was one here in Colombia that found almost 30% of the species to be threatened. And in 2016, in Brazil, they didn't find any difference with 2008. And now we have new data, new species, and we have a better way to calculate the distributional range of these crabs. And we also did this assessment because according to the IUCN Red List, all assessments on the Red List become officially out of date after 10 years. So the one that was made in 2008, it was out of date. So what has changed since 2008? We have new 5 genera described and new 44 species. We have 5 resurrected species. We have 7 taxonomic change, and we have 6 tiny names species. So now how we do it? So how you do what Red List assessment? So first we have to record all the information about crabs. So where they are, how they are doing, their ecology, everything you can know about it. So we did that review and we talked with some people working on freshwater crabs. And our major problem is that we know of crabs is just that they exist. Like only the taxonomy and kind of their distribution. So we don't know a lot about the ecology or about threats. So we have to work with little information, but you gotta work with what you have. So we map the distribution of these almost 300 species, and we did the red listing process. So according to the IUCN, in this process, we can have extinct species. We have 3 categories for threatened species that I guess you have all seen. We can have near threatened species, less concerning species. We have data efficient species that is very important for our results. Data efficient species means that we don't know how they are doing, so they can be extinct, they can be less concerned, they can be endangered, but we don't have enough data to know what's happening. So that's tricky because we can have a species that is doing really well, but we don't know, or we can have a species that is doing really bad, but we don't know. So for conservation actions, it's really hard to have data efficient species, but it happens. So the IUCN Red List used 5 criteria to categorize species in extinction. So the first one is population size reduction. The second one is the geographical range. The third one is the population size. The fourth one is also about population size and distribution. And the last one, some special analysis with population data. So if you remember, we don't have information about population or ecology of these crabs. So we have to keep it to the geographical distribution. This is the only information that we have. So according to the IUCN, there is some threshold. So here are thresholds. And if a species is below that threshold, we can suspect they are threatened by extinction. So if we suspect because of their geographical range that they are threatened, we also have to look at these other criteria according to the IUCN. So they need to be in less than 10 locations, that is different from localities. We have to look for the quality of the habitat, if it is declining, if it's gonna be declined, or if something happened in the past, but it hasn't get better now. And another kind of situations like climate change or extreme weather and stuff like that. So what we found? So as I told you, we don't have information. So the majority of the species that were threatened are threatened under criteria P, and some special cases under criteria D. So we don't have population data, but criteria D2 says that if the area of occupancy as is below 20 square kilometers, it goes under vulnerable D. That's the rule. So we don't have any critical endangered species, but we have 28 evaluated as endangered, 34 evaluated as vulnerable. So that make up 62 species threatened by extinction. We have 26 as more threatened, 55, as less concerned. And that's what I told you, that efficient species was important because half of the species we found it as data efficient. So the threats that we found are affecting freshwater crabs are, the first one is deforestation. So Pseudothelphusoidea depend on forest cover. First, because streams health depends on the forest cover, but also because, as I told you, crabs live also in land, but they cannot live out of the forest. So for the land part of their life, they need the forest. The second one that we have is urbanization because of the impervious surface syndrome. So what happens is that rivers are not being rivers itself, they are getting more channel. So that is awful for freshwater fauna. And also because if you remember, I told you they are distributed in mountain ranges. And here in Latin America, big cities are associated to mountains too. So the population growth in Latin America is next to mountains. So that's making it worse. The third threat that we found is mining, because it's connected with deforestation, water pollution. And here in Latin America, we have a lot of illegal mining, so the water pollution is worse. But the most important part is that heavy metals affect the pH in the water, and that affects the calcification process in freshwater crop. The other one that we have is dam construction, because it's connected again with deforestation, with water pollution, with loss in forest connectivity, and with loss in river connectivity. And the last one is invasive species. So here for the neotropics, we have especially the red plum crayfish and the big club crayfish, that they generate direct competition with freshwater crabs. So we think that they are displacing freshwater crabs, they affect the water quality, and they change the freshwater community too. So by country, what we found is out of the 16 countries that we have with Pseudothelphusoidea, 9 of them have at least one species under a threatened category. So the best ones are Mexico, Venezuela, and Panama. Suriname, Costa Rica, Ecuador, and Haiti have between 10 to 25% of the fauna in a threatened category, Guatemala around 30%, and the worst case is Colombia. Almost half of our fauna, of our Pseudothelphusoidea fauna, is threatened by extinction. And as I told you, we have the most biodiversity here in Colombia. So that percentage of threatened species here means that 15% of the total Pseudothelphusoidea fauna is threatened by extinction. So by general, we found 3 genera to be the most affected. That is Neotropical, with almost 70% of its species to be present, Ecologist with the 31%, and Freshwater with the 41%. So to highlight here is these 2 genera, Neotropical and Freshwater, are the more mountain freshwater crabs. They are really doing it up to the mountains, in the caves, so that's why they are more affected. And what difference we found between the first assessment in 2008 and now? So the main difference is the increase in the number of endangered species and new threatened species, because we have new threats, so, and we know a little more about crabs, so they are doing worse. And the data efficient species are still being a lot. So in 2008, they made some assumptions about what happened with data efficient species. So the first assumption was the best case scenario, meaning that all data efficient species will be okay. They're not going to be in a non-threatened category. We have a second assumption that says that the data efficient species will be threatened at the same rate as non-data efficient species. So for example, in 2008, 15% of the species were threatened by extinction. So that means that out of these 140 species, they will be 15% threatened by extinction. That will make up to 34% of the species threatened. And the third assumption is that all the species will be threatened, that will be the worst case scenario. So when we compare 2008 with now, we have like around 12% of the data species are now threatened. So that's close to the second assumption, meaning that if we know more about data efficient species, we'll have 43% of the species to be threatened with extinction. That's almost half of the Pseudothelphusoidea fauna. But what happened with data efficient species? So it's not that there are no crabs are there, maybe they are doing, but not. So here is an example of iNaturalist, is a citizen science platform. So all the red points are observations by community. So crabs are there, they are getting noticed, people know about them, but the problem is that researchers don't know about them. So what happened? The majority of the work that has been done in freshwater crabs here in Latin America have been focused on taxonomy, so describing new species, and that's it. That I'm not saying that's not important, but we need more than that. So we know what is there, but we need more information. So as an example, I'm gonna show you all the people that is working in freshwater crabs here in Latin America. So we have a person in the Caribbean islands. We have one research group with 2 professors in Mexico. We have 1 research in Central America and 2 professors in South America. And that's it. And the big problem is that they begin to work around the 80s or 90s. And they are really close to retirement. And we don't have people following their work. So we are not many people working. So we are going to have a generational gap. For example, here in Colombia, from Marta Campos, it's me, and that's it. So it's quite a work. So we need more people working on freshwater crabs. So we also calculate the red list index. That is a way to show the trend in the overall extinction risk. So if you see here, when you do 2 assessments, you can compare the red list index. So here is for birds, for mammals, for amphibians, for cycads, and for corals, and now for Pseudothelphusoidea. So if you see the overall trend, like we know amphibians are doing bad, but here it doesn't seem too bad. So here we can see that the decline in Pseudothelphusoidea fauna is almost the same as in coral reefs. So we are losing the crab pretty much. So what we can conclude is deforestation is our principal threat, and the main cause of deforestation are cattle ranching, agriculture, and drug trafficking. We have metal mining and dam construction as enhancers of deforestation and with high effects on macroinvertebrate communities. We have that human population growth and climate change are enhancers of those threats. Well, as I told you, we have 21% of the species under a threatened category, and half of them categorized as valuable fishing species. Since 2008, the situation has worsened, so we need conservation actions and we need more research, especially in ecology and population studies, to know what's going on. And to finish, I want you to remember that even we are talking about climate change and crude emissions and global, we are not talking about freshwater environments, and freshwater is what we depend on. So freshwater crabs can work as an umbrella species. So if we protect freshwater crabs, we're indirectly protecting the forest, the river system, and the freshwater fauna, and therefore we are securing our freshwater resources, that is water itself. So that will be it. Oh, okay. If you want to know a little more about, we published that this year in an Encyclopedia of Conservation of the paper. So here is the article if you want to know a little more, or you can follow me. And that's it. Thank you.
All right, fantastic talk by Al. I definitely learned a lot more about crabs than I knew before. So any questions for Al? Frank has a question.
Yes. Thanks a lot for very interesting presentation and just enlightening us about freshwater crabs. I mean, I've been thinking aloud in terms of, data is actually a very important, availability of data is actually a very important factor in studies like this when it comes to conservation and all of that. So how does the availability of data, or what is the quality of the data that was used, and how do you think that the availability or non-availability has actually affected maybe here and there the outcomes of your results? I mean, I'm just being a little bit curious here. Thanks.
Okay, so the IUCN has like a little is precautious about it. So when you do all the assessments, you have to be sure that what are you doing is accord with your data. So when you don't have enough data, you extrapolate it to the quality of the habitat. So we know freshwater crabs depend on the quality of water and forest. So if we don't have forest and we have polluted waters, it's gonna be like a bad suitable habitat. But if we don't have enough information, that goes directly to data deficient species. That's why we have half of the species as data deficient, because we just don't know. So the quality of the data really affects the assessment. So it goes to data deficient species. So in order to not affect the quality of the assessments, the option is data deficient. So if we have enough quality data, you do the assessment. You can do it, but if not, it's data deficient. That's why our information is saying that it's data deficient, but we know that crabs are not doing that well, but we don't have enough quality data to say it, to put it in the IUCN term. So I don't know if that answered your question or not.
Yes, of course. Thanks a lot, Ada.
Sure.
Any other questions for Ada or for Frank? Haley.
Well, I have one for Frank.
Okay.
So maybe you don't know, or maybe you know, but the macroinvertebrates that you have found, they are like good quality indicators or not? Like, I don't know, you are getting larvae mosquitoes in the plastics, or maybe you are getting like good macroinvertebrates.
Generally, I mean, it all depends on what you mean by, I mean, I'm struggling to understand what you mean by good quality data. I mean, generally, microinvertebrates are good water quality indicator here in, I mean, most of the water quality assessment studies that are done in South Africa actually utilizes macroinvertebrates. So, I mean, what is being increasingly seen from the data we are getting is that the sensitive macroinvertebrates, which are the EPTs, are actually showing some kind of negative correlation with plastic substrates. And again, I think we then have to, in our subsequent analysis, to start looking into unpacking the reason why some of these sensitive macroinvertebrates are actually showing some kind of negative correlation with plastic substrate. And yes, I mean, I think those are, I mean, good indication of quality of the water, if they have that answers your question.
Yes, I was asking because here in South America, sometimes macroinvertebrates are not good quality indicators depending on the family or the trophic level, but I don't know how that works in South Africa. So yes, answer my question.
Oh, okay.
So I can answer the chat questions, right?
Yeah, so we have two questions that are sort of similar in the chat from Haley and Brittany. So kind of going on the citizen science approach, and if this could possibly help to bridge some of these gaps.
Yes, so in general terms, yes, but in specific terms, no. So yes, because we know where the crabs are. So I have found some species that are weird, or I have one species that we don't know what it is because of our citizen science observation, but we look it up in the field and we didn't find it. But it's like a light for us about where to look. And it's also important because we know people are noticing them. So when you work with local communities, we have a different approach because we know they already know about crabs. The problem is that with photos, it's really hard to go through a species. So we need to look at the reproductive male system. So that's impossible with a photo. So we don't have the specificity of the species. But for sure, it's really important. It's really important. And it's nice because I'm curator in Naturalist. So I can talk to people and say, yes, crabs are important, tell me more about it. If you know or see, take a photo and tell me, and I look a researcher in the area to look it for, so it really helps. And I think it helps, maybe not in terms of the IUCN thing, but it helps in conservation. Because it doesn't matter if people doesn't know this is that species that is distributed, I don't know, from this point to this point that is in danger, but they know about the fauna that is in their river system and they can know the importance, so they can just conserve it just because it's a crab. That's it.
All right, thanks. So we're kind of at the end of our web session. If you guys have more questions for Frank or Ada, feel free to follow up with them offline because we're towards the end of our session. But before we leave, I wanted to remind you that if you're interested in presenting your talk in the Amplifying Voices webinar, I put the link in the application in the chat, so apply and you might be featured in next month or the coming months webinars. So the next webinar will be in December 8th, combining December and November together. So stay in touch to get the next links for the following webinars. And thank you so much today to Frank and Andrew and Ala for sharing your work with us today. And have a wonderful day wherever you might be.
All right, thank you, and bye-bye.
Thank you.
Thank you.